My name is Jeff Smith, and I'm the technical lead for the agroecology projects in the program. Webinar 2 is all about nematodes and their relationship with soilborne diseases, and is presented to us today by Lincoln University Senior Lecturer, Dr. Manjula Kularathna, coming to us today all the way from Sri Lanka, where he is currently working. In terms of the mechanics of the webinar, please submit questions at any time using the Q&A function in the Microsoft Teams toolbar. Like the question to help promote it up the list, so it'll be asked sooner. And if the Q&A is not available to you, please email questions to the news@alightetouch.co.nz email. So it's my pleasure to introduce Dr. Manjula Kularathna. Manjula's research aims to understand the detrimental effects caused by plant-parasitic nematodes, PPNs, and find new ways to protect crops. Manjula brings together knowledge from soil sciences, agronomy, plant pathology, and precision agriculture to create site-specific strategies for managing PPNs. Manjula's ultimate goals are to reduce crop damage, help growers get the most from their crops, and raise awareness about the problems caused by PPNs. So a good, very early morning to you, Manjula. I think you're 6 hours behind us, and I'll hand over to you. Thank you very much.
All right, thanks, Jeff. And good morning, everyone. And it's a pleasure to be here today. Even though I can't see your faces, I'm pretty sure that most of you are really excited to learn a little bit about the nematodes, right? Before we get into the subject, I would like to give like a brief outline of my academic career, just to as an icebreaker. So I did my bachelor's degree at the University of Colombo, Sri Lanka, and then decided to do my postgraduate master's and PhD in nematology at Louisiana State University in the US. So after finishing my postgrad degree, I joined Lincoln University in 2018 as a nematology and plant pathology lecturer. Next slide, please. Right, so back into the subject. You might be, some of you might be a bit familiar with the nematodes, but just to give you an analogy about these nematodes, for an example, if you remove all the matter out of the earth, except for the nematodes, the earth would still be visible through space due to this high abundance of the nematodes. So nematode itself, they are the most highly abundant multicellular organisms in the world. Right. Next slide, please. Right. So this map shows the distribution and the densities of the nematodes across all continents in the world. And the color scheme, which goes from yellow to dark red, indicates the nematode numbers per 100 grams of soil. So what I want you to focus is New Zealand. So here you can see we have a very high density of nematodes, and we have a very high diversity as well. So we have a big chunk of nematodes, but the knowledge we have on these nematodes is very, very limited. So if you look at the references, we haven't done much work on these nematodes for almost 2 to 3 decades. So there are a lot of catching up to do to understand these nematodes a little bit better. Yeah, so moving on. Right. So the question that I normally get when I talk about nematodes, people tend to misunderstand them with the earthworms. To give you an example, so the nematodes are in a different group, a different phyla than the earthworms, and the nematodes can be divided into 4 different groups, to aquatic, free-living, plant, and animal pathogens. So majority of the nematodes are in the aquatic environments, and around 25% live in the on the surface of the surface. But out of the whole nematode phylum, only 25% represent the parasite, the animal, and then the plant parasites. Next slide. Right. Just to give you an overview of the aquatic nematodes, we haven't done much of research on these aquatic nematodes. We know that they can be very helpful for the aquatic ecosystems. They can be parasites and also feeding on other microorganisms such as bacteria and maybe fungi. But other than that, we don't know much about these aquatic nematodes. But next slide, please. But when you look at the free living nematodes, we know a lot about these free living nematodes. So these are the nematodes which are not going to cause any damage to either animals or plants, but they are very, very important for the overall health of the ecosystem. So they can be divided into bacterial feeders, fungal feeders, predators, or just nematodes feeding on organic decaying material. So to give you an example of how important they are, we actually use them for our benefit for research in both medical and ecological studies. So to give you an example, next slide. Right. So you might heard about the forest restoration programs. It's a very common theme in New Zealand as well. So if you have an abandoned farmland that you want to convert into a native forest, you do these forest restoration programs. So what we find in nematodes, right, when you have an agricultural land, you have very high populations of these plant pathogenic nematodes. But in a natural environment, like a native forest, you find this free living type of nematodes more in abundance. So what we can use is the information by looking at the ratio of free living versus the plant pathogens, we can actually tell the health of the soil. To give you an example, in a forest restoration program, you start with a land which has been used for agricultural purposes for quite some time, and then you will have very high number of the plant pathogenic nematodes. But when you plant some native trees and then you let the ecosystem take over, you can see the number of plant pathogenic nematodes get lower, whereas the free living nematodes get in high number. So just by looking at the ratio of these two types of nematodes, we can actually tell the health level of a soil. So we use this as a bioindicator to tell the health status of a soil. So that's a good thing that we can get out of these free living nematodes. The next one, please. Right. So in medical research, we use a nematode. We call it as C. elegans. It's a model organism, which we pretty much know everything about this nematode. We know their genome and we know how this gene functions. To give you an example, how this is very important for overall human health. So typically, if something happens to our nerve cells, if it get damaged, the humans we can't regenerate the nerve cells. But when it comes to nematodes, especially these C. elegans, if something happens to their nerve cells, they can actually regenerate the nerve cells. So scientists are trying to understand the mechanism behind that so that we can use that information to cure nerve damage. So for an example, if you have like an injury to your back, you might get paralyzed because of the nerves getting damaged. But if you can understand how to recover that by looking at an organism such as C. elegans, we can actually use that for our benefit. So it's been studied a lot across the world in a lot of health research. Right. So the animal parasitic nematodes, I'm not going to go into detail. So if you have a livestock, you might heard about the nematode damage on animals. But in several parts of the world, around 50 to 60 countries are heavily infected by this nematode damage, especially on humans. So there are different diseases caused by these nematodes. We call them the intestinal worms, and sometimes they can cause these permanent disability due to the nematode damage. So in this example, you are looking at a disease called elephantitis. So the nematodes are injected through mosquitoes to younger children, and you can't see any symptoms until they reach the maturity or puberty. So what they do, the nematodes will go into the bloodstream and then end up in the, can go back one more slide, please? Thank you. And they go into the lymphatic vessels of the humans and they block all these lymphatic vessels. So the symptoms normally occur when they reach puberty, but when you see the symptoms, you can't actually reverse back the damage. So the only option would be amputation. So that's one of the things that these nematodes are capable of. But today's topic is more about the plant pathogenic nematodes, and we'll continue our talk only on plant pathogenic nematodes from now on. Right. So the plant pathogenic nematodes are microscopic. They are less than 1 millimeter in length, and most of them are in the soil. So you can't actually see them through your naked eyes. So the difference between all the other nematodes versus the plant pathogens, now they have to feed on plant cells, which has a cell wall. So now they are adapted, having a stylet, a pin, like a needle-like structure, so that they can penetrate the cell walls and absorb all the cell content. So they have a direct life cycle because, as I mentioned earlier, when I talked about the animal pathogenic nematodes, sometimes they need another alternate host or a vector to transport the nematode from one place to the other. But when it comes to plant pathogens, they don't need any of those. They can continue the life cycle within one plant. So they need a living plant for their survival, so they are known as obligate parasites. So those are some of the characteristic features of all the plant pathogenic nematodes. Right, next slide, please. Okay, so this is a good example to show the difference between two types of nematodes. So the one on your left side is a predatory nematode, a free-living nematode, which normally feeds on other nematodes, right? So if you look at the mouth region, the top bit of their head, you can see a big opening or a mouth opening on the predatory nematodes. So what happens when it comes to plant pathogen, all of this mouth lining would get together to make this structure a stylet, which is now a needle shaped structure which they use to penetrate the cell walls and absorb all the cell content. So if you're trying to identify a plant pathogenic nematode, this is one of the main characteristics that we normally look for. Next slide, please. Right. So in this slide, not sure whether if you click on the picture there, it's a small video in it. I'm not sure whether you can do it from, yeah, there you go. So here you can see these nematodes hatching out from the eggs, and they use this stylet to penetrate the egg shell. And on the other video next to it, you can see how they penetrate the cell walls and then absorb all the cellular content. So here you are looking at a fungal feeding nematode, which also contains a tiny stylet to penetrate the fungal cell walls. Right. Okay, so if you look at the economic damage and the losses, right? Pretty much all the plants that we grow are susceptible to either one species or multiple genera of plant pathogenic nematodes. So if you look at the menu, you can see pretty much everything that we grow is susceptible for these nematodes. So if you look at the economic losses, it's been said around 100 to 150 billion US dollars is lost annually. And depending on the amount of research that you are doing on that, the economic value, because the figure that you're seeing here is pretty much outdated, because the people who are working in this area is very, very limited. So with the current figures, we know that more than $150 billion is lost annually due to these nematodes. But unfortunately, because we haven't done a lot of work in New Zealand, we don't know how much that we are losing uh, uh, in New Zealand due to this nematode damage. So that's what we are trying to understand in our research at Lincoln University. Right. Next slide. Right. So the damage symptoms. So typically, if you look at the above ground symptoms, the symptoms that these nematodes cause are pretty much identical to nutrient deficiencies, soil compaction, and any other biotic diseases such as fungi, bacterial diseases. So you can't tell them apart which causes which when you look at the above ground symptoms. But when it comes to nematode damage, they normally tend to cause this patchy appearance in a field because nematodes are not evenly distributed in a field, and there are pockets in the field where we have very high numbers of the nematodes. So when you have very high numbers, they feed on the root systems, and then the plant would look very unhealthy in that part of the field. So if you have a nematode issue, you normally see this patchy appearance in the field. But if you want to properly see the damage, you need to look at the root system. So here on your left side, the bottom picture shows you two wheat plants, one infected with the nematodes and then the other without the nematode infection. So you can see a very, can you go back, please? Right. On your left bottom picture, you can see a root system with very high, very big root mass compared to a very unhealthy looking root mass as well. So when it comes to horticultural crops, carrot in this example, you can see this, what do you call, abnormal shapes of these carrots. And also in potatoes, you can see this blistering happening on the surface of the potatoes. So these things are not marketable, even though they are perfectly fine to eat, but they are not marketable, and it's the value of the crop would get reduced due to these damages. And on your right side, you can see a tomato root system infected by the nematodes, and you can't see much of the root system anymore due to the damage. And next to it is a healthy root system without any nematode damage. So the bottom line here is if you think that you have a nematode issue, you need to look at the root system to see if you can see any visible symptoms due to the nematodes. But if you want to properly understand if these symptoms are related to nematode damage, the best option is to send a soil sample to test for the nematodes. Right. Slide. Right. So let's look at the association with other soil-borne pathogens. So they have a direct correlation with soil-borne pathogens, especially the fungi and bacteria, because these nematodes make physical damages to the root system. So what it does, it open up the what do you call open areas for bacteria and the fungi to go in. And also because of this damage, there are lots of root exudates coming out of the plant as well. So that attracts more of these bacteria and fungi. So when you have nematode damage, you normally see secondary infection by fungi and bacteria. So good examples would be Rhizoctonia and Fusarium infection together with the nematodes. So what happens when you have both of these nematodes and another pathogen together, it makes things much worse than them being occurring independently. So apart from that, nematodes can carry viruses as well. So what you're looking at is viruses that's been transported by the nematodes, especially the grapevine fan leaf virus and then tomato ring spot viruses can be transported by the nematodes. So they are virus vectors as well. So those are some of the associations that they have with other microorganisms. Right. Next slide. Right, so I'm just going to briefly go through some of the research that we have been doing at Lincoln University, just to give you an understanding of what we know so far about these nematodes in New Zealand. So let's look at some of the work that we done in horticultural sector. Right. Slide, please. One more. Right. So when we first started this journey, we wanted to see what type of a damage that these nematodes can cause to the horticultural crops. So we used tomato as like an indicator plant because tomato is a very, very susceptible plant for nematodes. So we wanted to test the commercially available varieties to see which ones are susceptible and which ones are resistant. So after doing multiple greenhouse trials with the commercial varieties, we have tested pretty much around 15 to 20 different varieties. What we found is that all the tomato varieties commercially available are highly susceptible except for the grape type tomatoes. So what you see on the top picture on your right side, you are looking at a root system uh, with and without the nematodes. So when you don't have the nematodes, you have a very healthy looking root system. But when you have the nematodes attacking the root system, you are going to end up with a root system which can't uptake nutrients and water. So typically in a greenhouse setting, if you have this infection within several weeks, these plants would look very, very unhealthy, and eventually it will die out because it can't outcompete the damage caused by the nematodes. But on the bottom right picture, what you are looking at is a cherry type tomato, which seems to be very resistant to these nematodes. And if you compare the root systems with and without the nematode, you can't see any difference. So what we found from this variety trial is that these grape type tomatoes that we have commercially available in New Zealand is somewhat resistant to the nematodes compared to the other types of tomatoes. Right. Next slide. Thank you. And what last year we did a thorough survey across New Zealand, especially the Kumara growing regions. And what we wanted to understand is if these nematodes have a tendency to cause damage to our Kumara industry or not. So we did a survey across different regions, and then what we found is that we have nematodes in the soil, but when you look at the numbers, it seems like they're not going to cause any damage at the moment. Having said that, the sampling was done soon after the hurricane happens in Dargaville and Kaitaia. Therefore, what we are seeing here might not actually represent or not tell us the actual story. So what we are planning to do is to continue another set of surveys in a due course, and then we can relate whether these numbers can actually cause any damage to our Kumbara industry or not. So if you look at sweet potatoes in Australia, their threshold levels or the number of nematodes per kilogram of soil is way lower than what we are seeing in this graph. So their nematologist indicates that even like 20 to 30 nematodes per kilogram of soil can cause a significant yield reduction in Australian sweet potatoes or kumara. So if that's the case, we might have an issue with our kumara industry due to the nematodes, but that need to be tested because our environmental conditions and our soils are different than what's in Australia. So that's some of the work that we have done so far in the horticultural sector. Right, next slide. Right. So if you, the next one that I'm going to talk about is the cereal nematodes. So we spent quite a bit of time understanding the impact of these nematodes on cereal crops for the last couple of years. So we worked on wheat and maize mainly. So if you go one more, please. Right. So what we found, if you look at the literature, so we are looking at the surveys that we have done on maize industry. So if you look at the literature in New Zealand, we couldn't find any new literature or any new research done for the last few decades. So at the moment, we know that the nematodes are there in the soil, but we have no idea what type of an economic impact that they cause in the maize industry. So what we did, next slide, please. Right. So we did a very thorough survey across all maize growing regions in New Zealand. And what we found is that we have a variety of different nematodes associated with maize industry. But out of several different genera, one tend to be the most dominant one across all maize growing regions. That's the root lesion nematode. So if you look at the map in different regions, it's their abundance or their occurrence is very, very high across all maize growing regions. So next one, please. Right. So what we found 5 different species of this root lesion nematode associated with maize, and out of which the first one, Pratylenchus neglectus and Pratylenchus thornii, tend to be most damaging in maize industry elsewhere in the world. So we are finding those species in New Zealand as well. So if you go next slide, please. Right. And on the other thing that we found in pretty much all the fields, that these nematodes are not evenly distributed, as I mentioned earlier. So what you're looking at here is a mazed field, and the size of these squares indicates the population density of the nematodes. So the smaller colored squares indicates very low numbers per kilogram of soil. But if you have a very like a large square, that means we have a very high population of the nematodes. If you look at the scale up on your left side, you can see it indicates the number of root lesion nematodes per kilogram of soil. And in very high areas of the field, you can see around 15,000 nematodes per kilogram of soil. Because we haven't worked on this area, we don't know our threshold level in New Zealand. But if you compare the threshold levels elsewhere in the world, uh, people say around 2,000 nematodes per kilogram of soil can reduce the yield up to 30 to 40%. So if you look at the numbers that we have find here, we can see in some parts of the field, we have way higher numbers than the listed threshold levels elsewhere in the world. So that indicates that these nematodes might have an economic impact on these maize crops. So what we did, we wanted to test our hybrid lines, which is available to New Zealand as well. If you move on to the next slide. And we want to test the susceptibility of these existing hybrid lines that we normally grow in New Zealand. So we joined forces with Pioneer Seeds, and they gave us 15 different hybrid lines to test against these nematodes. So in this graph, what you're looking at is the reproductive factor. That means if you add 100 nematodes, what would happen to that 100 nematodes, whether they increase in population or decrease in population. So if the reproductive value is more than 1, that means they can reproduce very rapidly in that plant. So here you can see our 15 different hybrid lines. And if you look at the reproductive factor, you can see pretty much in every tested hybrid line, the reproductive value is more than 50. That means pretty much all the varieties that we have tested is highly susceptible to the Platylencus nematodes. Right. So far, we found that they are highly abundant in our fields, and also the hybrid lines that commercially available are highly susceptible. So when you put them together, that indicates there's a high risk of uh, damage caused by these Platylencus nematodes in our maize industry. So if we correct this issue, that might increase our yields significantly. So that's what we are trying to do in our next stages of the research. Right, next one, please. Right, so we looked at some of the issues that we have, and then let's now focus on some of the management strategies that we can use with our modern technological, modern technology that's available for us. Right, so if you go to the next slide, right. So back in the day, right, so if you find nematodes in a field, what we tend to do is to apply, if you are using a chemical to control them, we apply this chemical across the field. So typically, the sampling pattern would be doing like a big W across the field, and then mix them all together, mix all the soil together to make a composite sample. And if you find nematodes in the sample, you apply a chemical across the field. So that's what we have done doing so far. But next slide, please. But as we saw earlier in our maize example, these nematodes are not evenly distributed in the field. So if you do a proper sampling by dividing the field into small sections, then we can actually see where these nematodes are distributed, right? So that's what we normally do in our research. So we divide the field into small sections and then do a thorough sampling across the field. Right, next slide. And on the other hand, all of these soil-borne pathogens have a direct correlation between the soil physiochemical properties, such as soil texture, soil pH, and soil nutrients. So uh, there are technologies available at the moment so that we can detect the soil texture in a field. So what you're looking at here is a machine called Veris 3100 soil electrical conductivity mapping system. So what it does, it by sending an electric current into the soil, it can separate different soil textural classes in the field. So if you go to the next slide, by using that information together with the information from a satellite or GPS signal, what you can do is divide the field into different soil textural classes, as you can see in your picture one. So the darker colored areas normally tend to have very high levels of clay, whereas the lighter colored areas tend to have more of the sand in the field. With our nematological knowledge, we know that most of the nematodes tend to prefer this sandier part of the soil. So if you look at the second map, with our nematological understanding, we know that most of the nematodes are concentrated in the top bit of the field. And when you have this information with you, what you can do when it comes to the management, you can precisely manage the part that's needed the attention. So if you go to the next, uh, yeah, there you go. So by incorporating such technologies, what you can do, you can reduce the input that you're going to use. If you are using a chemical to control them, now you are going to use half of the chemicals that you used to use back in the day with our old methods. And also this will save money for the growers, and also it's very uh, beneficial for the environment because we are adding less amount of chemicals into the system. So this is called site-specific nematode management. And with our new technologies such as drones and then using different images, we can do similar things in this space. So that's some of the things that we currently use in order to manage these issues. And we are still developing our nematological research at the moment to better manage this nematode damage. Right. I think with that, these are some of the research that we have been involved in. And with that, I would like to conclude the talk and happy to take any questions. Thank you.
Great, thanks, Manjula. We have got a few questions coming through the Q&A.
Yep.
So I'll read those out to you. Given that nematodes are frequently associated with secondary infections, what approach would you recommend to quantify the impact of nematodes alone to effectively prioritise R&D investment, specifically targeting nematode-related biosecurity risks? Can you read all that? It's quite a long question.
I'll try to break it into small pieces. So the first one is when we have a pathogen together with the nematode, how can we correct the nematode issue? Is that the first bit of the question?
That's right, yeah.
Right. So the nematode itself, right? When you have nematodes in a field, we can't eliminate them from the field. So they are going to stay there for good. The only thing that we can do is to manage their population below the threshold level so that they are not going to cause any economic loss. So especially in a scenario like in New Zealand, we don't have a lot of chemicals available to control them, but we have options such as crop rotations and other methods to control these nematodes. So if you control the populations of nematode, they might still damage the plant, but the damage that they cause would not outcompete the plant can outcompete the damage of the nematodes because the population is very, very low in the soil. But when you have another pathogen in the field, even though the nematode population is low, they will still make these entry points for the other pathogen to get in. So therefore, you will still have an issue with the combination of the two pathogens, or the nematode and then the pathogen. So I think I hope that answers that question. The other one is the biosecurity risk. Is that, Jeff?
Yes. Yeah,
Right.
targeting nematode-related biosecurity risks.
Right. So we've been involved in one of the project with MPI because at the border, when you import like horticultural imports like bulbs, stems for horticultural purposes, we tend to use like a chemical treatment at the border. But because of the toxicity of these chemicals, the available chemicals are not available at the moment in the market. So we came up with a solution using a less toxic chemical plus a hot water treatment to remove all the nematode threats at the border before it comes into the country. So for our exports, we have the potato cyst nematodes currently as an issue, but we have a, a protocol to eliminate that issue before it goes out from the country. So we are keeping an eye of the threats that we might have in the future because with the climate change, all these tropical types of nematodes have a tendency to come into New Zealand and impact our crops. So we have a we keep a constant eye to check whether these nematodes are coming into the country or not.
Okay, great. Thank you. Next question is, what would explain the nematode distribution in the field on the slide you showed previously? And I think that was probably that maize field with the erratic
Yes.
establishment.
Yes. So to better understand the distribution, you need to do like a thorough survey across the field, right? So typically what we do, if you depend on the size of the field as well. So typically you need to take at least 10 to 15 soil cores within the field, but each soil core I mean, each sample is a collection of uh, 12 to 15 soil cores taken around a 4-meter radius. So it's a composite sample itself. So from one field, you have either 10 to 15 of these composite samples taken in different parts of the field. So that typically represent the distribution of the nematodes. So the sampling is the key to properly identify what's out there.
Right. And just thinking back to um, validating what you were saying about soil texture being an indicator of where nematodes would have higher populations. Did you validate that against actually testing the soil texture versus actual nematode counts?
Yes, so we've been working on this, especially when I did my postgraduate at Louisiana State University. That's one of the major research that we have been involved with. It depends on the type of species that you are dealing with, right? So for the root knot nematode, they have a very direct correlation between the soil texture and then their reproduction and pathogenicity. So if you're working with root knot nematodes, yes, there are multiple research articles already out there indicating the direct correlation between the nematodes and in the soil texture.
Great, thank you. I've got another question here on carrots. Can carrot virus Y be transmitted by nematode from old residual crop left in the soil to a new one just seeded?
Not that I'm aware of. So all the literature indicates because the nematode group that can transmit the virus, right? They tend to associate with the perennial crops such as grapes and then other horticultural crops. So their life cycle is around 8 months, that type of nematodes. And then during that 8 months, they can transmit the virus from one plant to the other. But for other crops, annual crops, we don't see this virus transmission through the nematodes. It's mainly with the perennial crops that we are seeing.
Cool. Root lesion nematode, what is the best cover crop to suppress them?
Right, right. So at the moment, we don't have a good answer for that. But if you look at Australia, they have different cover crops that they use to break the cycles of these nematodes that need to be tested in New Zealand just to confirm which one works best because their soil and their environmental conditions are a bit different than ours. So the cover, even though we use the same cover crop, we might not get the same effect. So at the moment, I don't have a clear answer for that. But as I recall, I'm sorry, I don't have a clear answer for that at the moment, but I can definitely send an email for the person who asked the question. I can look at it and then send an answer for that.
That would be great. We could add that to the webinars
Definitely.
when we post them. Yeah, thank you. I see Shandon's added to that question. How would you prioritise investments on nematodes if you don't know the actual impact caused by the nematodes?
That's a really good question. So at the moment in New Zealand, right? So we haven't done any research so far. So the only thing that we find, especially for the cereal crops, that we have a very high number of nematodes in our soil, and then pretty much most of the varieties that we grow are susceptible. So the next stage would be doing like a variety trial and then develop a threshold level specifically for New Zealand. So when we have that, then when you look at the soil sample from a field, we can compare the numbers that you're finding with our threshold level. So anything above that would definitely cause a significant reduction in the yields. So what we are planning to do, we are trying to join forces with FAR and then trying to develop this threshold level for the cereal crops, mainly for maize and wheat if we can. Then that would be your baseline. So you send us a sample, we look at the numbers, and if you have anything above the threshold, if you don't do anything to control that, you'll definitely lose like big chunk of your yields due to nematodes. So that's the next step that we are planning to do.
Thank you. Great, thanks. I hope that helps, Shandon. So I'm jumping over back and forth a little bit, but just going back to the root lesion nematodes, can they be washed to new places with excessive rain?
Oh, definitely, yes. So that's one of the main ways that they transport from one place to the other. Yes, definitely. And then the tractor tires or in all the sticky soil and everything, that's another means of transport.
Okay, cool. Got a few questions coming in. Have any nematode studies recently been conducted on outdoor herb plants within New Zealand? How would they be affected?
Not that I'm aware of. No, we haven't done anything. The only thing that we did was we looked at the bowling grains to see the impact of nematodes on cochula. But pretty much every place that we go is tend to have a big nematode issues pretty much everywhere we go. But other than that, we haven't done anything on any other crops, on any other plants.
Would you expect to see the same sorts of symptoms with that sort of ill thrift and poor establishment?
Uh, Probably. Because nematodes I mean, if you look at turf mainly, you will definitely see a lot of nematode damage elsewhere. So we might see something similar here as well, but that need to be tested as well.
Right, I think we're pretty good. I'll just refresh and make sure there's another one. What are the management options to rebalance nematode populations in a cropping soil? Management options to rebalance nematode populations.
Right. So the best option, so because we don't have a lot of chemicals to control them, the best option would be crop rotation. So that's been tested elsewhere. And sometimes the organic amendments tend to help as well. So typically, there's a group of scientists working in sweet potatoes in Australia. What they do, they use organic amendments before planting. So that tend to reduce the population significantly during the cropping season below the threshold level. So we have that, and then crop rotation in cereal crops that's already been tested in Australia as well. So we have few non-chemical options already available that we can adapt here.
So that'd be introducing compost, for example.
Yes, so they are using chicken manure and then sawdust, pine sawdust that tend to be working for them.
Yep. I think we're pretty good.
Okay.
Thank you, Manjula. It's great to see the focus on a not so well understood crop protection issue, and particularly your practical and solutions focused output. I think that's what our audience is really looking for. So appreciate your input there, and really interesting talk. Thank you very much.
Thank you so much. Yes.
Webinar 3, a focus on some of the key soilborne diseases, will be held on Tuesday the 23rd of September. and that starts at 10 a.m. So we look forward to you joining us for that one. Yeah, in the meantime, thanks again, Manjula, and have a good day.
Thank you so much. Thanks, everyone.